This adapted soil sampling protocol by Terravesta provides guidance for other CHCx3 project partners to assess soil physical properties, available nutrients, soil carbon, and soil microbiology. In short, this soil sampling protocol has three main objectives. First, to conduct baseline analyses using soil taken from 0 to 30 cm cores for sending to Yarra or land crop to analyse and quantify the following properties: soil texture, soil organic carbon, soil carbon using loss on ignition method, pH, macronutrient status, total mineralisable N, CO2 respiration bursts, and carbon to nitrogen ratio. Secondly, to conduct further soil analyses using soil taken from deeper depths of 30 to 60 cm for sending to Yarra or land crop to analyse and quantify the following soil properties: soil texture, soil organic carbon, soil carbon using loss on ignition method, pH, and bulk density. Then, finally, to conduct in-field assessments using soil taken from 0 to 30 cm soil cores, these assessments being visual evaluation of soil structure, worm counts, bulk density analysis, and local field notes or descriptions, otherwise known as soil habitat and characteristic analyses, these being things such as slope, aspect, altitude, relief, drainage, surface stoniness, and presence or absence of significant shading. Each field not exceeding 10 hectares is considered as a parcel to ensure manageable sampling and accurate representation. If the soil type, geology, topography, and management or crop history vary within the field, then one must separate said field into different parcels. For each distinct parcel, a minimum of 5 sampling plots should be selected to best cover that parcel. When selecting these sampling plots, one should avoid field boundaries, headlands, and gates or areas of traffic. One should also avoid sampling after heavy rainfall, as it's hard to remove soil cores out of the mud, after cultivation, as loose soil will influence the bulk density analysis, and after periods of drought, especially in heavy clay soils. When sampling, one requires the following equipment: a notebook or a pre-printed spreadsheet on a clipboard to record the sampling data, an indelible ink pen, a spade, a large white sheet, although we found cutting open a supermarket bag for life works well, a clean auger with a minimum length of 60 cm and ideally 30 mm diameter, and or a corer 40 cm long and 5 cm in diameter, a spatula, a clean bucket, a clean trowel, a tape measure or a 30 cm ruler, a stick, and optionally a 5 metre bit of string. This is to help mark out the plot area. Sealable sample bags provided by Yarra, and a mobile phone with GPS and what 3 words installed, as well as a camera if you are unable to take pictures on your mobile phone. The following label requirements are required for each sample: the sampler name, the target area, the plot number, the parcel number, the GPS and what 3 words reference, and the test code provided by Yarra, this being BSE SOL, and the date of sampling. For each of the 5 plots, the following samples are required: 3 0 to 30 cm cores for completing the VESS analyses and worm counts, 1 0 to 35 cm core to obtain 0 to 5 and 30 to 35 soil sections for the bulk density analysis, then 2 0 to 30 cm cores to be bulked and sent to Yarra or land crop, and 2 30 to 60 cm cores from the same locations used previously, which are again to be bulked and sent to Yarra or land crop. For each of the plots, we found it useful to mark the central point with a stick to gauge our whereabouts within each plot. This could then be used to attach the 5 metre length of string to map the perimeter of each plot, so we weren't sampling outside of it. In Terravesta's case for taking bulk density samples, we used a 40 cm split tube corer. This required us to use the full 40 cm of length in order to obtain the deeper 30 to 35 cm samples. Before coring, choose an appropriate location within 5 metres of the central plot point and record the location. Then prepare the soil by clearing the soil surface of any residues using a trowel, ensuring a level surface for accurate sampling. Then gently hammer the split tube core into the ground until it reaches the desired depth of 40 cm, ensuring it is level with the soil surface. Remove the core from the ground and carefully extract the soil core from the plastic film. Measure the core to check that it's greater or equal to 35 cm. Then lay a tape measure alongside the length of the core and cut 0 to 5 cm and 30 to 35 cm samples with a spatula, and then place these in the corresponding sample bag. Once finished, these samples can be sent to NIAB for bulk density analysis. After each core has been taken, it's essential to clean the equipment to avoid contamination between samples and different plots. To note, if trimming is difficult due to stones or roots, and it results in a depression within the sample, discard the sample and take another. Do not try to add extra soil to compensate for the depression. In Terravesta's case for the VESS analysis and worm counts, again, we used a 40 cm split tube corer. Firstly, record the What3 word location for each VES sample, then hammer the core 30 cm to the ground and remove by hand. Then cut off any extra soil below 30 cm and place the core onto a white sheet or tray. Place a ruler or tape measure on the soil block, take a picture and estimate the soil layer depths, assuming there's more than one layer. Assuming there is more than one layer, for each layer, break up the block into natural aggregates, keeping the structure in place, and take another photo. While doing so, count and record the number of adult and juvenile worms present in each layer. Then score the soil according to the VESS scorecard and repeat this process 2 more times within each plot. For the 2 0 to 30 cm baseline samples per plot, which are to be bulked and sent to Yarra or Landcrop, we again use a 40 cm split tube corer. As with all the samples, firstly record the What3 words at a randomly selected location within the 5 metre plot radius around the central marker. Hammer the core into the soil until it reaches a depth of 30 cm from the surface, remove it from the soil, and then remove any surface debris, roots, or large plant matter, but do not take out any stones or gravelly material. Check the core length at 30 cm and then place the soil core into a bucket. Then repeat the aforementioned steps for the second 0 to 30 cm sample in that plot. To note, it can be helpful to mark out the holes with sticks to easily find them for taking the 30 to 60 cm samples afterwards. Once all the 2 cores are in the bucket, thoroughly mix the cores using the spatula to break up the soil, then using the bulk sample, half fill the labelled sample bags ready for sending to Yarra or Landcrop. Finally, clean the equipment and repeat for the 2 30 to 60 cm samples in that parcel using the same holes, but this time using a 1 metre long auger to access the deeper samples.